Hello, everybody. Welcome back to another round of biology of cells and tissues. Today we're going to be talking about the urinary system. So we're going to try and get through all of this in one. It might get split up into two. As always, leave a like on the video if you can. Subscribe if you haven't already. And let's dive right in. So today we'll be talking about the urinary system. Some of our learning objectives: functions and components, structure and histology of the kidney, blood vessels, blood flow, structure and histology of the nephron, as well as the structure histology of the ureters, bladders, and the male and the female urethra. So here's kind of our overall urinary system. We're going to start with kind of the functions of everything, as then dive into the kidneys. So our functions, we're going to be filtering all of this stuff. We filter out the metabolic waste. We're filtering the blood. We maintain our acid-base balance. We maintain fluid, electrolyte levels, as well as we regulate blood pressure. The big things that we're going to focus on, we have the kidneys, the paired kidneys, one on each side, paired ureters, the bladder, and then the urethra. Most of this presentation is the kidneys. The kidneys are often, I'll be frank, one of the least favorite things to learn in this program because they seem to be a little bit more complicated. So I really took some time to break a lot of this down to make sure that you guys get a good solid base for kidneys, because you're going to see a lot more of them in the future. In physiology 1 and 2, you're going to talk about the kidneys. Gross anatomy 2, you talk a lot about the kidneys, and then in pathology courses as well, kidneys come up a lot. So some of our main structures that we look at: first, the hilum, the cortex, the medulla, medullary rays, and then renal lobes. So here is our hilum. We have the cortex on the outside, medulla on the inside. These are the ones we can't really see in these pictures, but I have others for that. So the hilum specifically refers to a bundle, or also could mean a notch. So on the medial, there's a concave medial region of the kidney. There's this notch here. This is the hilum, just like in the lung, how there's the hilum where everything goes in. Same thing here. We get the blood vessels, the lymphatics, the nerves, all that kind of stuff. Specifically, the renal artery enters, renal vein exits, and then the lymphatic vessels and nerves also. Nerves enter and lymphatic vessels exit here. Within the hilum, there is a series of structures where the urine is collected. These are the renal pelvis, major and minor calyces. This is just a quick definition of what they are. Basically, they're like these little cups. So see right here, as we filter it out, we're going to circle at the very end, towards the end of this presentation, we circle all the way back to this. So what I just want to point out is within the cortex and medulla, we're going to be doing all of our filtering. And then that filtrate comes all the way down and it comes into the minor calyces, which those then all converge into a major calyx, and then all of the major calyxes merge together and become the renal pelvis. We're going to circle back to this later, so I just want to introduce it there because it's one of the big structures. So the kidney is divided into two chunks. We say there's the cortex and then there's the medulla. So the outer region of the kidney is the cortex, and that houses the renal corpuscles. This is where we where we can talk about nephrons and where filtration, all that kind of stuff happens. We have a whole section coming up about the nephrons and about the renal corpuscle, so we're going to hold off on that until we get to that section. But understand all of that is happening in the cortex. Next, the cortical columns. So we notice how these darker spaces here, these are the medulla, and then these spaces in between where it's lighter colored, all of that is the cortex. So the cortical columns are these portions diving in. They're considered an extension of the cortex. Now, the medulla, it has the pyramids of the tubules, which we'll get into exactly what all of that means more when we talk about the nephrons and stuff. We call these structures right here, we call them the medullary pyramids. They have something called a papillary duct. We're going to get to that again in the way distant future towards the end of the PowerPoint. We'll talk about more of what that is. And then those cortical columns separate out the different pyramids. And I'm actually going to get rid of that down there. The cortical columns are technically part of the cortex, so we just want to make sure, but we need to know that they're separated by the cortical columns. I'm going to unhighlight that here so we don't get too confused. Okay, there's our picture, there's our cortex. These are the medulla. The pyramid is inside of the renal medulla, and the renal columns are separating the medullas. Medullary rays. This is kind of difficult to visualize. However, this is really kind of more vocab definition than anything. Okay. So the medullary rays, these are columns of tubules. When we get to the nephrons are split up into two sections. They have the renus corpuscle, and then the renal corpuscle, and then the tubules. So it will come full circle about this. So the tubules of the nephrons extend from the medulla into the cortex. So from these pyramids into the cortex. The medullary rays contain portions of the loop of Henle and collecting ducts that transport urine from the nephrons to the medulla for further processing. That's going to make more sense in a little bit when we talk about the nephrons. And then division of stuff, we have these renal lobes. If you take a medullary pyramid, the overlying cortex, and the associated renal columns, that all kind of gets lumped together. So if we were to draw one, we could say right here, more like this, this is a renal lobe. There's a slightly different one that's slightly smaller. If we just have the medullary ray plus the cortical labyrinth stuff going on, that's the renal lobule. We don't really talk about that in this specific course. We do mention, though, the renal lobe. So those are the big general structures of the kidney. We're gonna get into, this is probably one of the more confusing portions. It's the blood supply of the kidney. I really tried to break this down so that we can understand it a little bit easier. So it all starts with the descending aorta. Our heart pumps blood. It goes up the ascending aorta, the arch of the aorta, and then down the descending aorta, down almost a midline, just a little bit off to the left. And then when it gets down towards the area of the kidneys, the renal arteries branch directly off of the descending aorta. So we start off, we have the renal arteries come off of the descending aorta. The renal arteries then branch into smaller arteries that we call segmental arteries. I should back up one quick sec. The renal arteries enter the hilum of the kidney. Then it splits, and you get segmental arteries. And then arteries do what they're going to do. They split again. And when they split again, they're running along the sides of the medulla. And those are interlobar arteries. And both these arteries are going to wrap around the top of the medulla, and the part where they get towards the top, what I would call the top, you could say the more external part of the medulla, the more superficial part of the medulla, then they are called the arcuate arteries. Now it doesn't stop there. The arcuate arteries give off more of these little branches. You see these little branches popping off here along. These are more arteries, and we give them a name. And actually, we give them two names because they have an AKA. These are our cortical radiate arteries. They're also known as interlobular arteries. Okay, don't get those two confused. Interlobar arteries are on the sides, on the lateral sides of the medulla. The interlobar artery is coming off of the arcuate arteries, and they are going through the cortex. Okay? We continue this whole process. All of these points, these are arteries. If we think back to our blood vessels, arteries get smaller and smaller and smaller until they eventually become arterioles. Same thing here. The interlobular artery has an arterial, it branches, it splits off, and an arterial that we call the afferent arteriole comes off of the interlobular artery, aka that cortical radiate artery, and that's going to go into the structure called the renal corpuscle, which is where filtration is going to happen. We're not going to talk about filtration just yet, but that's where it's going to happen. Remember, afferent, that word afferent, the A means to or towards, so it's going towards the renal corpuscle. That afferent arteriole, after arterioles we get capillaries, so the arteriole splits into a web of capillaries inside of that renal corpuscle, and this web, though sometimes they'll call it a tuft of capillaries, that is called the glomerulus. Okay? They're going to separate all across, loop around, and eventually, just like they come all back together. So capillaries will split into like a capillary bed. If you remember, think about the blood vessels. And so then they spread all the way out, and then they eventually converge again. Same thing here. They spread all out, make up the glomerulus, and they come back and converge again. And they leave the renal corpuscle in the efferent arteriole. Okay. After going through all of that, the efferent arteriole, that is the end of our arterial side of the blood flow. Now we're going to start heading back towards the heart, and this is now the venous side of things. So we have these two structures. We have peritubular capillaries, or we have the vasa recta. So depending on what type of nephron, we're going to talk about what the nephrons are in a little bit, what type of nephron that we have is going to determine which of these two is used on the venous side. If we are talking about a cortical nephron, so that nephron is up in the cortex, almost entirely in the cortex, that is going to use the peritubular capillaries. If the nephron dives deep down into the medulla, these are called juxtamedullary nephrons, so cortical nephrons, medullary nephrons. Those are going to use the vasa recta to start their venous side journey. Cool? Moving on. From there, it's going to continue kind of in that reverse order of naming, and it's going to follow its path back until it gets back out the renal vein, right back out of the hilum. With a tiny baby exception, from the peritubular capillaries or the vasa recta, whichever it's coming from, it'll either enter back into that cortical radiate vein, also known as the interlobular vein, and then go into the arcuate vein, or if it's closer to directly attached to the arcuate vein, it can skip the cortical radiate vein. Okay? That's a little bit more in depth than we necessarily need to know. If you get a specific question about it, okay. If not, just know it goes in the reverse order. After we go through either the paratubular capillaries or the vasa recta, we're going to go reverse order. We go into the interlobular veins, then drain into the arcuate veins, and we just continue back from there. The interlobar vein, then the renal vein, and eventually the inferior vena cava, however you want to pronounce it is fine. Okay, so that is our forward and backwards of all of that. Hopefully, I did a decent enough job of breaking all of that down. I know that can be a little bit confusing, especially with all the different names, and this is very often tested kind of heavily. So we want to make sure that we know this really well. Here's just another picture. I kind of broke this picture down a couple of times to give us the different components that we want. This is the picture as a whole. This is a really good thing to sit here, maybe remove all of the names of stuff and try and write them in yourself and practice this a couple of times until you get the hang of all of the names and the order that they go in. Clinically, I think that this clinical application should be a little bit later on, actually, when we talk about the glomerulus. However, this is kind of related to blood, so that's why it's put here. So the glomerulus, which is that web of capillaries, can get inflamed, and that is called glomerulonephritis. It can either be acute or it can be chronic, but it usually stems from a humoral immune reaction. The humoral immune system is that immune system where those B lymphocytes create antibodies. Okay? So what can happen is you can get deposition or kind of like depositing or getting stuck the antibody antigen complex. So when the antibodies find the thing that it wants to attack, it binds to them, and then a bunch more antibodies come and they all kind of surround it and attack it. So it becomes like this ball, and we call that an antibody antigen complex. Those can essentially get stuck inside of the glomeruli, which is the plural of glomerulus. Or as well, circulating antibodies can actually bind to glomerular antigens themselves, which is more of kind of like an autoimmune type situation, or any other unrelated or extraneous antigens that may have also been stuck in the glomeruli for whatever reason. Essentially, the accumulation of these immune complexes then causes a local inflammatory response that we call glomerulonephritis. Depending on whether this is, it's due to some type of allergen, or if it's actually due to more like an infection, you'll see different things coming through the urine and the filtrate, which you will learn a lot more about in future courses, especially like in your systems pathology course, as well as the lab diagnostic courses. You'll cover a lot of that stuff. Okay, now the meat and potatoes, the nephron of the kidney. There's a lot of information here. We're going to break it down and make it as painless as possible. And I've tried to highlight the specific things that most commonly pop up on questions, but again, all of this is going to be testable material. So I shouldn't say all of it. I've added a good fair bit of stuff that is just going to help with understanding. So if it's highlighted, if it's bolded, pay extra special attention to those things. And then hopefully through all of it, you can help understand all of it. So the nephron is the basic structural and functional unit of the kidney. Professors love to ask about the functional unit of stuff. So always know that is. And the nephron is responsible for all of those functions that we mentioned earlier that actually we want the kidney to do. Each kidney contains between 1 and 4 million nephrons. And there's two main parts of the nephron. We have the renal corpuscle, which is where we do blood filtration, and then the renal tubule, which is where we process that filtrate, getting it down into urine and either reabsorbing the stuff that we want to keep or releasing, secreting all the stuff that we don't want. So first we're going to talk about the renal corpuscle. Again, this is the site of blood filtration. We're going to talk about the glomerulus, Bowman's capsule, podocytes, mesangial cells, and then something called the filtration barrier. So the glomerulus, we already kind of mentioned what this is. This is a capillary tuft or a bundle made up of fenestrated capillaries. If you remember what that word means, fenestrated is one of our types of capillaries that have lots of small pores or little holes in it. Not quite as many openings as sinusoid capillaries, but much more space than our continuous capillaries. So they're fenestrated. The blood comes in through the afferent arteriole and then exits through the efferent arteriole, and it allows for small molecules to pass through as well as fluid to go through while holding on to all the big stuff. The Bowman's capsule, this is the structure that encloses that glomerulus or the and as well as the urinary space. The urinary space is just the space between the glomerulus and the Bowman's capsule, so all of this, the empty space, that's where the urine is going to be, the initial filtrate is going to be taken out, and they call that the urinary space. Bowman's capsule is made up, it has two layers. It has an outer layer and the inner layer. The inner layer is the visceral, and that's made up of these cells called podocytes. And then the parietal layer is the outer layer, and it's just a simple squamous epithelium. So right here we have podocytes on the inside, and then simple squamous epithelium on the outside. Now, the podocytes we want to talk about specifically, even though they're part of the Bowman's capsule, we kind of differentiate them as their own thing because of their role that they play. So these are the cells of the visceral layer of the Bowman's capsule. Their job, though, is to wrap around the glomerular capillaries. And the way that they do that is they have these things called pedicels, or pedicels, however you want to pronounce it. These are long, finger-like projections that come off of those podocyte cells, and they interdigitate. This is like interdigitate, kind of like this. They wrap around and grab on to the capillaries to form a filtration barrier. And then between these pedicels, we get these filtration slits. This will make more sense when we look at the picture. So here's the Bowman's capsule again. Here are the podocytes. So imagine we take right here, these podocyte, and then we in this capillary right here, and we zoom in right there. This coming through is our fenestrated capillary. And there's its endothelium, there's that basement membrane, all that kind of stuff. These structures here, so all this white that's here, these are our podocytes. Podocyte, 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 all through out here. And they give off these processes called these pedicels. I just want to zoom in pretty good. So these are our pedicels, and see how they kind of lock together almost like fingers, kind of like this. They interdigitate right there. So they're grabbing on and wrapping around the capillaries. And in between these little processes, they have these slits. Those are those filtration slits. That's where the filtrate is able to escape through and come out of. Here's more of an electron microscope view of what those podocytes look like. We're going to come back and talk about those filtration slits and the pedicels when we talk about that filtration barrier just in a couple slides. Finally, we have mesangial cells. These are located between the glomerular capillaries and within the connective tissue framework known as the mesangium. These are our structural support cells. They support the capillary walls as well as the basement membrane, and they act as phagocytes. So if there's any kind of debris, yucky stuff going on, these guys swallow it up, make sure everything stays nice and clean. Okay, the filtration barrier. We say that the filtration barrier, this is a combination of all of the different structures, which ensures a selective filtration of blood plasma. Because we want to make sure that we keep the right things inside of our blood and filter out the stuff that we don't want in our blood. So we have 3 specific things that help us make sure we're filtering out the right things, and they create this filtration barrier. So the endothelium of the glomerular capillaries, those are those fenestrated capillaries that allow plasma to come through and some solutes, but block the bigger structures like the red blood cells and the platelets. The fused basal lamina. This is the basal lamina is shared by the endothelial cells and the podocytes. So this is a selective layer that prevents large molecules like proteins from passing through. You don't need to know all of these super specifics. Know what's highlighted on this slide. However, I have this other information in here so that you can better understand what's going on. Finally, the pedicels of the podocytes. These create those filtration slits that we showed, which again further restrict, they make this barrier stronger, and they restrict the passage based on the size of the things trying to be filtered out, as well as the electrical charge of them. Here's what that barrier looks like. We have endothelium, we have the basal lamina, and we have pedicels. This picture again, the basal lamina, endothelium, and the pedicels. Okay, we're going to go ahead and end this video right here. This is going to be part 1, and then we're going to do the other half of the nephron and the other structures in part 2. Okay, thank you so much for watching. Good luck studying, and have a wonderful day.